Well, thank you for the invitation. It's great to connect virtually, even though we can't connect in person. I was looking forward to enjoying some of the nice weather down in Southern California, but maybe another time. So today I'll talk about some work that's been going on in my lab, really since the beginning of my lab. And we've always been focused on what is the first part here, which is bioengineering early CNS morphogenesis, taking organoid technology and trying to make it something that can be biomanufactured, reproducible, and as closely mimetic of the human system as possible. And then we've made some progress in that, which has allowed us to start applying the early progress we've made. And that is to create a tool that can be used for looking at neural tube defect risk, and I'll talk about what those are, as well as we started to look at developmental neurotoxicity as an assay with that tool as well. So before I begin, I have a conflict of interest. I'm new to this. We just started this startup in March. And so I'm new to doing these type of slides, but I'm basically the interim manager for a startup, which is trying to commercialize the Rosetta Ray technology that's going to be presented in this talk. So the human central nervous system, I'm not sure the audience and how familiar everybody is with CNS development. So I'm going to start with sort of a broad overview, very 300 level view of CNS development, enough to make you dangerous at understanding what I'm talking about. But the central nervous system is really thought to be one of the most complex tissues in the human body, and some say most complex piece of matter in the entire solar system. Looking at just sort of gray cell sagittal slices of the CNS, you can see as you go through the brain, through the spinal cord, there are many different regions of this tissue. So it's not one homogeneous structure. It's actually a conglomerate of many different tissues. They're all precisely connected to one another. And that connection gives you the functional output of the central nervous system. And if you look at any one particular small region of the CNS, you'll find that the cells that are in that anatomical region are specific to that region. And the cytoarchitecture of that tissue is specific to that actual tissue, allowing it to have the contribution to the overall CNS function. Now, this regionality of the central nervous system shows up primarily in disease for sort of the lay audience. So there are known diseases that you commonly have heard of that are region specific. For example, Parkinson's disorder. So you have the main target cell type, which are dopaminergic neurons, a specific type of neuron within the central nervous system. And you have clusters of these at all different regions. So these is A9, A8, A10, various different clusters of dopaminergic neurons throughout the forebrain and midbrain. However, the disease really targets the A9 substantia nigra cluster, and the understanding as to why that occurs is not well understood. But you really have decimation of the dopaminergic population in this area, and that leads to the issues or symptoms of Parkinson's disorder. The same can be said for other neurodegenerative disorders, such as amyotrophic lateral sclerosis, where you have the target cell type in that disease is the motor neuron. These are the sole pathway that the central nervous system connects with the rest of the body. And you have motor neurons that project from all regions of the hindbrain through the spinal cord to various tissues of the body. And in that disease, really the ones that project to the limbs are the ones that degenerate first. And in a lot of patients, the motor neuron connections that project to other regions such as the eye, as well as the gut system, those persist throughout the end stages of the disease. And it's not well understood as to why there's difference in the pathology in different types of motor neurons. So this regionality is very interesting and sort of highlights the diversity of cell and tissue types within the central nervous system, the eye, the brain, and the spinal cord. And when we use iPS cells, that is reprogramming somatic cells using a genetic trick that's well known now, in order to redifferentiate them into various cell types, if we want to see the pathology of that disorder, we actually need to make the neuronal or glial cell types that are specific to various regions of the CNS that are affected. Otherwise, we don't see the same type of disease phenotype. And this is useful for creating high-throughput screening models to discover therapeutics that could potentially ameliorate the pathology of these disorders. Also, you could try to, and people are, deriving the cell types that are lost in degenerative disorders and transplanting them back into the patients in order to try to improve their situation. Now, overall, this works well, but when you're trying to model diseases using iPSCs, you have some inherent limitations. And that is that while we can differentiate the cells to various regions of the CNS, the context in which they exist within the dish is not biomimetic. So standard cell culture on hard plastic substrates is not biomimetic. It lacks the cell phenotype diversity. So for example, a transverse section of your spinal cord is roughly the size of your thumbnail. And so you can imagine that fits pretty well within a 12-well plate. So within that tissue slice, you would have anywhere from 10 to 20 different cell types. And so you can imagine that they all sort of talk to each other. And so you're lacking that diversity within normal tissue culture dishes. And the cytoarchitecture of the culture, of course, is not mimetic. And also you have a very immature phenotype. So it's an ongoing question of how we can age these cell types properly. So in comes neuroorganoid technology. And really, this is the cutting edge technology that we use in the academic sphere and it's the best model we have for mimicking development of the human organism. And in the central nervous system, we can create various types of neural organoids. By culturing pluripotent stem cells as an aggregate, you can immerse them in an extracellular matrix hydrogel. You don't have to, but you can. And then culture them in a stir tank bioreactor. And the convective flow within the bioreactor causes interstitial flow within the organoid itself, allowing for transfer of nutrients and oxygen that allows the organoid to grow to millimeters inside with limited hypoxia that occurs within the center. You still get some, but it's limited and allows large growth of this aggregate. And what you see when you look inside of that tissue is that the cells, when guided down the neural lineage, they start to spontaneously try to recapitulate developmental aspects. You can get formation of these sort of ventricular stem zones that have layering of neurons on top of them. You can get all 6 neuronal layers of the cerebral cortex. You can get all different layers of the optic cup or the retina. And if you allow those organoids to grow to very large sizes, then you can get various different tissues within the same organoid. And the organization of those tissues is quite somewhat random, but you do get a diversity of tissues that form. And this is sort of highlighted in this particular slide, where within a neural organoid, for example, you can have multiple brain regions that form within that organoid. And if you guide your neural stem cells to different head-to-tail regions or rostrocaudal regions of the central nervous system axis, you can generate cortical, midbrain, cerebellar, and spinal organoids as well. So the system is quite powerful for trying to mimic development of the central nervous system. And at the micro scale, the structures that form within these organoids do very closely mimic the structures that form within normal CNS development. However, at the macro scale, because of the spontaneous morphogenesis that occurs within these tissues outside of the body, so it's not receiving those cues that really orchestrate this morphogenesis, the tissue is very basically a mess in terms of its spatial organization. It doesn't recapitulate sort of the spatial patterning that you see within the developing central nervous system or the developing neural tube, which is the analog for the central nervous system. And so this is a limitation. It is a limitation that while you have the human genome and human background within these organoids, you don't have the same type of tissue structural organization that you get within an actual model organism. This limits our ability to study various aspects of neurodevelopment, circuit level physiology and behavior, looking at things like psychiatric disorders, as well as aging continues to be an issue. So my lab sort of looked at this very early on when Yoshiki Sasai was just publishing his organoid protocols in the late 2000, about 2008. And we said, well, is there a way that we could apply engineering principles to make this a more reproducible experimental platform? And so while the central nervous system is thought to be very complex, the actual quote is that it's the most organized piece of matter in the solar system. And this is from Scott F. Gilbert, a very notable neurodevelopmental biologist. And what that means is that we can use, the decades to century worth of neurodevelopmental biology in order to understand the players that are required to provide those cues to the developing organoid and try to create a roadmap for how that is done, and then try to implement that roadmap in a engineer system, or as engineers we can make it. So how does the CNS develop? So in a very brief description, the central nervous system starts as a tube of cells. So you're looking at the chick embryo here. My hand's a chick embryo. You're looking at the top down. So this is the neural tube. You have the head region, the midbrain region, and the spinal cord you can see down here. If you take a transverse section of that embryo and then look head on, you can see the neurotube structure here. If you stain it and zoom in, you can see that it's basically made up of a single cell layer of neural stem cells. We call these neuroepithelial cells. And each cell has 1 foot on this apical portion. This will become your ventricles and the lumen of your spinal canal. And also 1 foot on this basal portion. This is where they deposit the extracellular matrix to create their basal membrane, and this will persist throughout the development of the CNS. So very early on, the CNS sort of walls itself off from other regions of the developing embryo. Now, within this neural tube structure, the diversity of cell types and ultimately the tissues that are generated arises by diversifying the neural stem cells at the outset of neural tube formation. So in a very sort of coarse way, we can describe this as sort of a 2-axis system, the head to tail axis or rostrocaudal axis, as well as the dorsal ventral axis or back to stomach axis. And depending on where neural stem cells lie within this neural tube along these 2 axes, they get patterned with various transcription factors that essentially diversify their differentiation trajectory and give rise to the immense level of neuronal and glial diversity that exists within the central nervous system. Ultimately, depending on where you are along that neural tube, those neural stem cells are specified to give rise to particular progeny, and those progeny then migrate radially to expand the neural tube, giving rise to brain and spinal cord and retina tissue structures. And depending on the specification of the neural stem cells and the progeny they create, you get different tissue cytoarchitectures that form and ultimately link together to give you the function of the central nervous system. So that was a very brief overview of the CNS, but it informs sort of our approach to trying to recapitulate CNS morphogenesis in a dish. And ultimately, we'd like to do this in a very controlled format using engineered culture systems to apply these various cues that can regulate morphogenesis in a controlled manner. And so we've broken it up into sort of 4 different parts. First, we like to be able to generate these single neural tube-like structures. Second, we'd like to be able to have radial expansion of our tissue as the neural stem cells within that neuroepithelial layer give rise to progeny that then migrate radially. We'd also like to be able to pattern our neural stem cells along those 2 major axes, dorsal ventral and rostrocaudal axis. And then eventually the central nervous system will get large enough that you will need vascularization. So this is one thing that sort of is a nuance, is that the central nervous system, when it first forms and a lot of this morphogenetic patterning and morphogenesis is occurring, it's largely avascular. But eventually it reaches a size where you need to have the blood vessels come in. And so in standard culture, you're limited in being able to recapitulate these different aspects of morphogenesis. What we're trying to do is apply or integrate biomaterials, engineered biomaterials with pluripotent stem cells so that we can control and reproducibly create these different aspects of morphogenesis. And today, while we've made progress in all these areas, I'm just going to talk about one particular area, and this is the area where we made our first progress, which is being able to take standard neurostem cell derivation protocols that give you sort of a plate full of these rosette structures, is what we call this in vitro culture, that mimic the polarization and shape of the neural cross-section of the neural tube, and control the induction of these structures such that we can get spatial and temporal control over where they occur in our dish and put them in an arrayed format. And we've actually started now to apply that tool to try to look at neurological disorders. So first, we had to generate neural stem cells. Being engineers, we wanted to do this in a controlled manner, defining as much as the culture system as we could. So when I first got to Wisconsin, James Thompson, his group had figured out how to derive pluripotent stem cells using chemically defined media, as well as a chemically defined culture substrate. We basically took that system and said, well, how can we differentiate our cells into the neuroepithelial cells or the neural stem cells? And we tried sort of basic protocols, a control condition, which just removes the factors that maintain pluripotency, as well as inhibition of single SMAD and dual SMAD. These were two protocols that were published at that time. Dual SMAD inhibition is widely used as well. What we saw when we did this is that all of our experimental groups seemed to very easily cause the cells to differentiate into PAX6 positive, SOX2 positive neural stem cell cultures in a very homogeneous manner. So PAX6 is a transcription factor that's deterministic for defining the neural stem cell phenotype at this early time point in differentiation. And you can see we can get plates full of these rosette-like structures that form this polarized incadherin, which is a cell-to-cell contact ligand and receptor. And so this was very surprising to us. Turns out that when we dug into this a little further, the defined conditions in which you maintain the pluripotent stem cells allows this transition to occur without the need to inhibit any type of SMAD signaling that can be caused by TGF-beta or BMP proteins or factors that are in your media. And so that was interesting, but now we had a very defined way to generate forebrain neural stem cells. And so when we differentiate them, they form these wells full of rosettes that form spontaneously. They do this in 2D as well as in 3D. And we basically said, well, what's different about our cultures that limits us from getting a single rosette? And we looked at the size scale of the neural tube when it forms and made a very simple hypothesis. Well, what if we take our culture and shrink it down to the same size scale? Can we then induce single rosette formation in order to mimic a single transverse slice of the developing neural tube? So in order to do that, we use tried and true technology developed by George Whitesides in order to micropattern self-assembled monolayers of alkane thiols on gold substrates. The alkane thiol, the sulfhydryl group on that molecule, spontaneously forms a bond with pristine gold, and the alkyl groups through van der Waals interactions align themselves, and now your surface chemistry is defined by whatever the omega group is on the end of the alkane thiol. You can make that omega group a polymerization initiator and make this substrate even more stable by grafting polyethylene glycol brushes from the surface. Polyethylene glycol is the most non-fouling material that we have to date. And so by doing that, you can make your surface essentially non-fouling, meaning that it won't absorb proteins and therefore won't allow cells to adhere to it. Now, this is useful because you can combine the formation of your self-assembled monolayers with microcontact printing in order to make microscale patterns of your self-assembled monolayer and subsequently your grafted polyethylene glycol brush on your substrate. And so here we have 300 micron circle array where we have polyethylene glycol that is grown. We've used 3D FTIR in order to show that we have the PEG brushes in those specific regions. And if you reverse that pattern such that you have PEG brushes everywhere except for those 300 micron diameter circular arrays, then you can seed or coat the surface with an extracellular matrix protein such as laminin or matrigel. It will only adsorb where the polyethylene glycol brush is not, and then when you seed cells onto it, they will only attach in the areas where you do not have the polyethylene glycol brush. And this gives you a well-defined surface for controlling where the cells will adhere and ultimately the morphology of the footprint of the tissue that you will create. Even in culture media where you have high serum proteins, this platform can still maintain the control over cell adhesion and micropatterning for upwards of 2 months, actually. So this gave us a substrate that we could control the morphology of, or at least the footprint of the tissue that we were creating in our culture dishes down to the 100 to 500 micron scale or length scale at which early CNS development occurs. So we then went back and looked at our differentiation protocol, saw that we started to get PAX6 induction, as shown here in the red, by day 3, really started to have polarization occur with incadherin around day 4. And so we did a simple experiment of taking our cells at day 4 and seeding them onto the micropattern culture substrate and just culturing them out for different periods of time to see if we could reform these polarized neuroepithelial structures. Indeed, this does occur actually about 5 days after after you seeded the cells onto the micropattern substrate, you then got formation of these rosette tissues. At the 300 micron diameter circular micropattern, we saw that we could actually generate single rosettes, but we could only do this about 30% of the time in that particular experiment. And so we wanted to see if we could increase the efficiency of generating these singularly polarized neurorosette tissues. And so we tried seeding at different time points along the differentiation protocol. And we saw that no matter when we seeded the cells on day 2 or day 6, after 5 days, they basically form these polarized neuroepithelial structures on top of the micropattern regions. So that led us to our first sort of protocol, which was that basically after 5 days of seeding your differentiating neural culture, you can generate SOX2 PAC6 neuroepithelial tissues with high density. And our question became, is there a morphology or a geometry in which we can engineer this micropattern such that it would give us 90 or upwards of 80 to 90% efficiency in generating these singularly polarized rosette structures that we could use as models of a transverse slice of the neural tube. And so we did what any good engineer would and just tried sort of a spectrum of shapes and sizes corresponding to the surface area of the circular micropatterns at 200, 300, and 400 micron diameters. When we ran this experiment, we saw that across the board, as you decrease the size of the micropattern, you increased the number of micropatterns that gave you a singularly polarized rosette tissue. So the red here indicates the number or the percentage of micropatterns that had a single polarization, whereas the green represents the percentage that had more than one polarization, and the blue represents the percentage that did not have a polarization after 5 days of growth on these micropattern cultures. But the numbers here were too low to really differentiate between the shapes. So we partnered with a computer scientist, or a couple computer scientists actually, who do image processing and sort of large data machine learning algorithms, in order to make an algorithm that could take an image of an ink-adherent stained neuroepithelial tissue, identify the polarized ink-adherent regions, and then read out various parameters of those polarized regions to let us know if we had a single rosette, what was the size, and various morphological aspects of it. And doing this allowed us to increase our analysis by the order of magnitude and started to show that the circular micropatterns seem to be optimal at the 200 micron diameter scale in order to generate single rosettes. And so we then, when doing this, we were still only able to get about 40 to 50% of our rosettes to be the singly polarized neuroepithelium. And so we went back and tried one more perturbation to the protocol, which was either seeding at day 4 or seeding outright onto the micropattern substrate in order to conduct the entire neural induction on the micropattern substrate. And when we did this, we saw that seeding at day -1 essentially allowed more cells to accumulate on the micropatterns, and that led to a significant increase in the percentage of micropattern tissues that had a singly polarized cytoarchitecture, upwards of 80%. When we do this now, we're upwards of 90% on average. And this seems to be corresponding to the fact that you have more cells on the substrate when you seed the pluripotent stem cells at day 1 versus not. And what I've been showing you up until now in all these images are confocal slices, so you can see the polarized ring quite well. But what I want to make sure you understand is we seed these microarrays as a monolayer, but very quickly, because they're proliferative cells, they can't grow in the XY direction, so they grow in the Z direction. And they become hemispherical disc, where the polarized ink-adherent creates basically a vesicle inside of that hemispherical disc. So you have sort of a mini organoid that has formed on these micropatterned arrays. So up until now, I've been talking mostly about forebrain neural stem cells. Now, as I mentioned before, the CNS is quite diverse. And so there are many different regions along the rostrocaudal axis where the neural tube forms. And so we wanted to see if we could look at other regions along that rostrocaudal axis, particularly in the spinal cord region, in order to see if we could do the same type of micropattern singular rosette emergence protocol. And the posterior central nervous system is quite diversified by collinear combinatorial expression of these Hox transcription factors. You have 39 of these genes in your human genome, and they come on and specify whether the neural stem cells are patterned to various regions of the hindbrain and the spinal cord, as shown here. That diversifies the differentiation trajectory to give rise to neuronal subtypes that only occur within those various regions. This is important because the Hox gene code basically is present in all bilateral species and is responsible for segmenting along the head to tail axis in all bilateral species up. And in particular in the human, it's responsible for mapping the projections of the central nervous system to various tissues of other germ layers. And if the Hoxes are deleted, for example, or mutated, then you have serious deformations that occur. If it's deleted in the fly, for example, then you delete entire body segments of the fly. So to make a very long story short, we basically came up with a program, a differentiation protocol, following what was known in developmental biology, particularly the theories of Kate Story over in the UK, in order to take pluripotent stem cells, differentiate them to a primitive streak cell type, which we call neuromesodermal progenitors. And as long as they're in the immediate conditions, Wnt, FGF, and GDF11, that allow maintenance of this cell type, they will start to express their Hox genes. As soon as you add on Wnt, they will start to express their Hox genes, and they will express them in a manner that is collinear and combinatorial, just like the axis extension that occurs within the normal growing embryo. And then we can differentiate them to neural stem cells or neuroepithelial cells by simply transitioning to a media that contains retinoic acid. And this allows us to create a spectrum of neural stem cells that regionally correspond to different rostrocaudal positions along the posterior central nervous system. So doing this, we decided to try to shoot for cervical spinal neuroepithelial cells, and we saw that we could micropattern them. We could get some singularly polarized rosettes. The staining here indicates that they are motor neuron progenitors to indicate that they are indeed in the spinal cord region. The Hox profiling shows that they express Hox genes that are in the spinal cord. But when we looked at the percentage of tissues that gave us singularly polarized rosettes, we saw that that percentage or the efficiency decreased significantly when we tried to micropattern them on 200 micron diameter circles. And this was perplexing to us because these were neural stem cells, just like the forebrain ones that we had generated, but they were neural stem cells that were regionalized to the posterior central nervous system or the spinal cord. And so frustrated, we simply went back and profiled all different sizes of circular patterns to see where the forebrain and spinal cord cells would preferentially give us a singularly polarized rosette structure. Now for the forebrain, we saw again that at the 200 and actually the 250 micron diameter circular patterns, that's the size they prefer to generate this singly polarized neurorosette structure. However, in the spinal cord tissues, they preferred much smaller micropatterns, down to 150 micron diameter micropatterns to get similar efficiencies in singular rosette formation. Now, this was perplexing, and so we said, well, is there a difference in the biomechanical properties of these cells, particularly the cell contractility properties that control rosette formation? So we basically added ROCK inhibitor to the culture, which would limit their ability to contract and form the polarized ring. When we did that, we saw that the micropattern size that the forebrain neuroepithelial cells preferred shifted to the left and decreased the size of micropattern that they would give us singly polarized rosettes on. And when we did that for the spinal cord cells, it completely abrogated their ability to form rosettes at any of the micropattern sizes that we tested. And so what this told us is that even though these are neural stem cells, all neural stem cell populations, their regionalization to the forebrain versus the spinal cord imparts a differential biomechanical characteristic to those cells and their ability to form these polarized rosette structures. And in a hand-waving fashion, this may be one of the reasons why the ventricles in your forebrain are much larger than the canal in your spinal cord. But this was just an interesting phenomenon that explained why we had to use different size micropatterns in order to create efficient singular rosette emergence using forebrain versus spinal neural stem cells. So having sort of shown that we can generate these, control the emergence of these rosette structures in order to get singly polarized rosette tissues. Essentially, now we have spatial temporal control over where these rosettes should form. We can show where they should form in our culture dish, and that predictive power should allow us to understand if that rosette formation is being disrupted in any way, shape, or form. And we're using this rosette formation as an analog to actual neural tube formation. So the question became, could we use sort of our bioengineered human neurorosette array, standardizing that process and looking at rosette formation along different portions of the rostrocaudal axis of the central nervous system in order to become a screening tool? And we think that could potentially be useful. The reason why we think it could be useful is because there are congenital birth defects that affect neural tube formation. And we know just from anecdotal evidence of doing this that neurorosette formation is very sensitive to factors that affect neural induction of human pluripotent stem cells, cell viability or proliferation of pluripotent stem cells or neural stem cells, proper biomechanical function of these cells, and any factors that will affect these sort of signaling pathways that are required to generate these cells and the rosettes that they form. So looking at the neural tube defect literature, and this is going to be closure of the developing neural plate. So it closes at the midbrain hindbrain barrier and sort of zips up to the forebrain and to the spinal cord. This occurs within the first 4 weeks post-conception, so sometimes even before people know they're pregnant. And this can go awry. You can have neurotube defects that occur at any point along the head to tail axis of the central nervous system. Now, when this occurs in the forebrain, it exposes the brain to amniotic fluid that causes degeneration of the tissue, and that is embryonic lethal. That particular disorder is called anencephaly. However, probably more well known amongst the lay public is when this occurs in the posterior central nervous system. This again causes degeneration of that tissue, but that is not embryonic lethal and leads to the disorder known as spinal bifida. Now, neurotube defects as a class are quite prevalent. They're the second most prevalent leading congenital malformation, only second to heart defects. So this is a useful disorder to try to go after and understand better why this occurs. Now, the only known prophylactic treatment for preventing a neurotube defect is by using periconceptional folic acid treatment. So if you get pregnant, you go to your doctor, they're going to prescribe to you daily supplementation of folic acid. And the reason for that is because in a clinical trial from mothers who were previously affected with neurotube defect pregnancies, it was shown that periconceptional folic acid dietary supplementation could reduce the reoccurrence of a neurotube defect in subsequent pregnancies by about 70%. So because of that study, actually, governments around the world have adopted fortification of their grain products with folic acid so that their entire population had folic acid supplementation. And this, as tracked by the CDC, caused a significant decrease in the rate of neurotube defects. However, that decrease is only about 30 to 40%. And so it's maintained or stabilized as still being a very prevalent congenital defect, affecting about 3,000 pregnancies per year in the United States alone. And that rate goes up as you go outside the United States. Now, this is a perplexing disorder because the factors that can cause neurotube defects can be genetic predisposition in the parents, as well as environmental factors. So there are numerous drugs that are FDA approved, EPA approved, that had to be restricted because they could cause an increased risk of neurotube defects. And there are also environmental toxins that we can be exposed to that can cause an increased risk for neurotube defects. So there's a complex interplay between genetic predisposition and these environmental factors that leads to neurotube defects. And because of that complexity, this is an ideal type of scenario for creating a screening tool that could be used to explore the etiology of neurotube defects. Now, in particular, as I mentioned before, there have been FDA-approved drugs, EPA-approved chemicals that were used that had to be either canceled or restricted because of their discovery that they increased the prevalence of neurotube defects in exposed populations. And this is, even still a contemporary issue. For example, the antiretroviral HIV therapy, dolutegravir, is known to cause an increase in neurotube defect risk. It's still used because of its effective effectiveness in preventing HIV, but it's not well, or it's still quite putative as to whether it has a direct effect on causing an increase in neurotube defects, despite the effect that was shown in epidemiological studies. And so a tool that can look and screen for factors that cause neurotube defects, both on the environmental and the genetic side, would be useful for regulatory decisions as well as drug development pipelines broadly. So we spent some time figuring out how to make this assay off the shelf, meaning that we could create frozen cell banks that we can validate for their ability to create, good neurorosette cultures. We could then seed those frozen cell banks directly onto our rosette array substrates, and in 6 days, they would form an array of neurorosettes. And you could expose them during that process to various chemicals and use a single image readout in order to figure out if you had affected cell viability, proliferation, morphogenetic patterning, neural induction, or some abnormality of the actual rosette that forms. And you could do this using automated image analysis pipelines. So this creates a scalable screening pipeline for using this tool in order to look at things like neurotube defects. So that's exactly what we have started to do. We started to basically run our neurorosette array, looking primarily at forebrain for now. We just got a grant to look at other regions of the central nervous system as well, exposing our developing neurorosette arrays to chemicals over the 5 days of the protocol. And we started off looking at dimethyl sulfoxide, simply because if you're doing drug library screening, they're typically dissolved in DMSO, so we need to make sure that our assay could perform at levels of DMSO that would be useful for screening. We looked at glycolic acid as a negative control, and then we looked at compounds such as valproic acid and methotrexate, which are used clinically but restricted for use for pregnant females because of their known risks for neurotube defects, and also a pesticide that has been correlated with the high occurrence of neurotube defects in the exposed populations. So when we looked at dimethyl sulfoxide, we got sort of dose response curves, and I'm just going to orient you here because the next couple of slides will look like this. On the far left side, we have cells per tissue and whether we have inhibition in the cell number per tissue at the end of the rosette formation process. The next graph is going to look at percent of cells that are PAX6. This allows us to examine whether our cells are being induced into a neural stem cell fate. Then we have the percentage of tissues that give us a singular rosette emergence, and we can get nice dose response curves there. And then we have an overlay of the 3 metrics to basically see if there is an increased sensitivity in one metric versus the other 2. So this is sort of our neurorosette array profile that we can generate. And as you can see here with DMSO, we can go up to 0.1% dilution of DMSO volume by volume, which is great because that means we can do 1,000-fold dilutions and still get good rosette formation on our assay. Interestingly enough, when we do exceed that, the rosette formation is what suffers first. It's statistically more sensitive than the other 2 metrics. Now, when we looked at glycolic acid, we saw no dose response, and so this was good because we shouldn't have one for just increasing the glycolic acid levels in our array. However, when we looked at valproic acid, which is a well-known histone deacetylase and well-known to cause an increased risk for neurotube defects when treating epilepsy patients or acute mania patients, we did see a nice dose response where we had increased in inhibition along all 3 parameters. Interestingly enough, the increase in the rosette formation inhibition metric essentially was the most sensitive aspect of our analysis, and this corresponded to sort of order of magnitude ranges that people have seen with other assays as well. Now with Benamil, we saw again nice dose response curves. It was quite toxic in terms of relative concentration in regards to valproic acid. We see similar levels as other people, but here we actually don't see an increase in specificity or in sensitivity to the rosette metric, basically indicating that this is potentially just killing cells. And that's causing 100% cell death, and that's leading to the lack of rosette formation. So different than valproic acid in its mechanism. Now, when we look at methotrexate, this is an anti-cancer drug. It's quite safe. We actually don't see 100% cell death. So it does inhibit cell death, but only up to about the 50% point. We don't see a significant effect on neural induction or differentiation, but we do see a dose response for rosette inhibition. And again, this is the most sensitive metric. And it goes to show that methotrexate actually is specifically inhibiting rosette formation prior to when it's having any other effects. And this is a well-known factor for methotrexate because it inhibits folic acid metabolism, which, as I showed before, was one of the more well-known pathways for preventing neurotube defect risk is to supplement patients with folic acid. So methotrexate has a very well-known pathway in inhibiting folic acid metabolism, which therefore causes increased neurotube defect risk. And here we see that is in causing an inhibition of single rosette formation at much lower concentrations than it causes any other effects. And so because there was such a specific pathway for methotrexate, we then went back to see if we could supplement with a folic acid substitute, which essentially acts downstream of methotrexate, or the enzyme that methotrexate inhibits in the folic acid pathway, in order to rescue the dose response that we saw. And indeed, as shown here in the circles, you have the untreated condition, and in the squares, you have the supplementation with 5-MTHFA. And you can see that we shift the curve to the right when we supplement with the 5-MTHFA, or the folic acid substitute, basically rescuing the effects that we see when increasing or exposing our arrays to methotrexate. So this is basically showing that our array is, or the effect of methotrexate on our array is happening through a very specific pathway analogous to what is known to occur in vivo. So we, of course, are starting to screen more and more compounds as we can get funding to do so that would show the utility of our assay for neurotube defects. And we also think that it might be useful for broad developmental neurotoxicity screening because the rosette formation process is so sensitive, at least in our hands, to perturbations from various chemicals that we put into the media. And so we have a ongoing collaboration with an agrochemical company where we've done some screening of various compounds, some that we added, which are shown in the white boxes here on the compound name. These are standard compounds that you would find in any sort of developmental neurotoxicity library. And we also have some that were added by the agrochemical company, and most of those are pesticides. And here we're showing that our array can, we did this at a single concentration level, so 10 micromolar, just because that was the amount of money we have for the project. But we're showing here that we can detect essentially hits for a number of these compounds, which allowed us to calculate a very sort of basic sensitivity and specificity level of 100% and 96% for our assay in this very small limited library. So those numbers will change as we continue to screen more and more compounds. But what this partnership allowed us to do is learn how to run our assay, which was great. We also saw that our assay, because we can get very high statistical numbers, so in each well of a well plate, we use a 12 well plate for now, we're scaling up to a 96 well plate, we can get basically 400 to 1,000 tissues in a 12 well plate. We only analyze 40 of those. So we have an N of 40 for each well, and then we do that in biological triplicate. So our statistics that we pull off of this array are super strong and allows our assay to be very sensitive to any perturbations in rosette formation. And so we have in the red here indicated sort of positive hits. That's where we see, a greater than 25% decrease in the metric that we're analyzing. Again, cell viability, neural differentiation, and single rosette emergence. We added another one for rosette area because that seemed to have an effect that was affected in tissues for compounds where we didn't necessarily see a decrease in single rosette as well. But we also saw that our assay could pick up very sensitive perturbations where we saw less than a 25% decrease, but it was still statistically significant due to the fact that we have such high numbers coming off of our array assay. And so we sort of indicated those as warnings because we wouldn't call those hits because there's such a small decrease, but they do indicate that potentially there is an effect there. And so basically from this, we saw that our assay in this very small screen has good sensitivity, good specificity for factors that affect the CNS development. For example, thalidomide is a very well-known developmental toxicant, but it actually doesn't have neurodevelopmental toxicity. It affects the skeletal system in its formation. And so we saw a warning sign here with cell viability at 10 micromolar, but we saw no effects on actual rosette formation. We also did compounds where because we were worried about the lack of metabolism in our assay, we simulated metabolism using the S9 liver fraction, doing just a simple predigestion before adding the compound to our assay. And we saw that for several compounds where we didn't see any effects, such as ciproconazole, such as propositamide, before metabolism, once we metabolize those compounds, we did see a direct effect on rosette formation and neurodifferentiation in the case of ciproconazole. Also, we saw factors like chlorpyrifos, which is a well-known neurodevelopmental toxicant, but is thought to inhibit neuronal processes or the firing and synapse formation of neurons. And we saw that actually gave us a slight hit in our assay prior to metabolism. But once we did the metabolism, gave us a significant hit at the neurorosette formation stage. And so that was surprising that for a compound that's thought to act much later in development, we could also see the toxicity in our early developmental assay. And we had other compounds like verapamil. This is the last one I'll highlight, which is known for its cardiotoxicity. It affects ion channels that are present in cardiac cells. And so it's flagged as a developmental toxicant, but it's not a developmental neurotoxicant because it doesn't affect neural pathways. So this is just some of the stuff that we're starting to learn about our assay. We think it's going to be very sensitive to developmental neurotoxins, particularly pointing out some neurotoxicity that maybe hadn't been seen before. This, again, is at a 10 micromolar level. So ideally, we like to do dose response curves to make sure that we're hitting physiologically relevant concentrations. For some of these, that's true. For other ones, it's not. And we have a number of compounds that just outright killed cells, as shown by the compounds that just having a significant effect on cell viability, but we couldn't measure anything else after that because it killed all the cells. Those, of course, need to be done at lower concentrations as well. So hopefully what I've shown you in this talk is that being able to bioengineer biomimetic and reproducible morphogenesis of organoids is quite powerful. In our particular case, we're looking at central nervous system or neural organoids, because that allows you to understand in a spatiotemporal manner how your cell culture should behave, how the tissue should form, and therefore, much like a model organism, such as a mouse, you can understand or analyze when that is perturbed. And that is very powerful for understanding and investigating the etiology of various diseases, as well as for toxicological aspects. And we plan to use this in a personalized medicine approach to discover preventative therapies that can be used, particularly with neurotube defects, as a prophylactic to prevent or decrease neurotube defect risk. Now, this was just the initial sort of success that we've had in what is likely going to be the outline of my lab for years to come of controlling neural organoid morphogenesis by being able to standardize rosette formation, being able to look at this at all different head-to-tail regions of the developing central nervous system. And we're not the only ones in this space. The lab of Ali Brimalu at Rockefeller with Eric Siga, they're also creating neuraloid models. They have gastroloid models as well. Zhaiping Fu at Michigan is creating embryoid models. So this field of sort of synthetic embryology is being birthed and should be very useful for looking at diseases in a human genomic and physiological context. And so with that, I'd like to thank all of our funding sources that have allowed us to do this work. The people in bold here have contributed to this work, and collaborators as well as former students and current students. I don't do anything in the lab anymore, so this is all their work. So I'd just like to thank them for that and our funding agencies. And with that, I will take questions.
That's great. Thanks so much, Randolph. Really quite elegant. While we're waiting, maybe I'll just pose a few few questions that occurred to me. It's a very elegant system, and as a developmental biologist, I find it intriguing to potentially watch development happen in front of your eyes. Apart from looking, as you have so far, at how the neural tube reacts to various perturbagens, have you followed this neural tube system through development? Let it continue to develop, and do you actually start seeing various kinds of subtypes of neurons develop, and then maybe even glial cells develop, and neural activity and circuitry start forming if you let it go beyond what you were talking about now?
Yeah, definitely. So on the micropattern platform that we're showing you here, if you just let them grow, they'll eventually become spheroidal enough where they'll just pop off. And so they'll make very uniform neuroorganoid spheroids. And if you let them grow out following standard protocols that are out there from Pasca and Noblish Lab, Lancaster, then they'll form standard neuroorganoids. You'll get the cadre of neuronal cells that form. If you're in the forebrain, you'll get the cortical neurons. You get the cadre of glial cells that arise, and they will start to have spontaneous activity within those cells. So yeah, we've done that just to make sure our cells were what we think they are.
Right. Will they extend all the way to start? Obviously, neurogenesis comes first, but then do you actually start seeing various glial cells start forming, just as you might see in development?
Yeah, after the neurogenesis phase, you get astrocytes or GFAP positive cells that start to form. The radioglia are there from the early on outset, but then the I guess standard astrocytes start to arise. If you take them out long enough, we actually haven't taken them out long enough because it's a 2 to 3 month protocol. You start to get oligodendrocytes, as other people have shown.
Right. And will you actually start seeing some lamination and also migration if you let it move out?
Yeah, so they refer to it as pseudostratification because when you get these rosettes that form within that aggregate, the progeny will migrate radially, and then you get sort of a layering of your neuronal subtypes. If you're doing corticogenesis, then you get standard corticogenesis in its inside-out manner that forms within these spheroids that you now have sort of floating around in your system. So the normal process of corticogenesis, when you go to other regions of the CNS, the temporal manner in which these cells arise, or the sequence, is largely recapitulated within these organoids.
Great. In addition to looking at molecular changes, molecular perturbations, and then obviously the neuroanatomical aspects, are you able to assay function in some way, either through calcium imaging or through even electrophysiology or things of that sort, MEA, or?
Yeah, so from an engineering aspect, that's very interesting. And so you can start to look at function as people like the Alison Mortry's lab at UCSD has done. Putting them on MEAs, you can use calcium imaging to look at the function. The question that we've always had is the physiological normalcy of that function. So if you aren't controlling very well the cytoarchitecture that forms, then the connections that form between these cells, how mimetic are those, and the actual signals that you're measuring is sort of an outline question in our head. Now, doing this in the 3D organoids that form is challenging, right? Because of their size and the post hoc analysis. We actually are trying to explore, making de novo slice cultures, essentially. And that would allow for interrogation of those tissues using standard focal microscopy. And so it could be tracked a little easier without some more labor post hoc analysis and methods. So that's a route that we're looking into versus trying to do everything in 3D, although we still have our projects in 3D as well, or in suspended 3D, shall I say.
Some questions are starting to now come in through the Q&A function, and Russ Henderson asks what he calls a very broad spectrum, I guess more philosophical question. And he was wondering do you envision doing any of this modeling in silico, basically envisioning tissue culture, tissue on chips, and then combining that with known signal transduction pathways and doing some neurocomputing to figure out what might be going on.
Yeah, this is fascinating. I don't have the skill set to do that, but others do for sure. For example, Tom Knudsen at the EPA, they have a whole division that is looking at synthetic embryology in silico. And so we have been talking with them. We actually had an EPA grant that sort of helped fund some of this work at Wisconsin, and have continued to talk and with Tom to try to combine that. That's exactly one of the things that we would like to do. That would require a partnership, and we're happy to partner with people to do that. The computational modeling side is not my forte, but we'd love to work with people to do that.
All right. Another question kind of speculates, could you possibly let your z-axis really take over so that you could start actually modeling the spinal cord or the spinal cord in development? Is that feasible?
So yes is the answer to that, but it's not the z-axis in the way the question is posed. So we have a very interesting finding that essentially, and I won't say how we do it, but I'll essentially say the result, which is we can biomanufacture neuroepithelial tubes. And when I say biomanufacture, I mean biomanufacture. And it builds from this micropatterning technology, essentially allowing them to go in the z-axis to a degree to the point where they actually close over and make a tube-like structure. And so it's interesting how that works, but we should be submitting a manuscript for that pretty shortly.
Well, that sounds pretty exciting. Have you ever tried to juxtapose your neural tube with, for example, other kinds of tissue, for example, muscle or cardiac or anything else, anything else to try to create Uber systems?
Yeah, so we have collaborated with people, particularly to add our cells to, for example, a motor neuron to skeletal muscle type model with Penny Gilbert up at Toronto. We're currently collaborating with Michael Moore at Tulane to connect dorsal root ganglia to our central nervous system model tissues. And so we're moving in that direction. It always has to be guided by a very good question to ask because those systems get complex, of course. But yeah, we partner regularly with others to try to fuse our tissues with other tissues that they would normally connect with in normal human development.
Have you ever made any of these mini neural tube models from, for example, IPS lines that come from patient or genetic diseases or from patients with diseases? And what do you see, if so?
Yeah, we haven't done that to a large extent. We have done iPSCs for these Rosetta arrays, and they largely work in the same fashion. You might have to tweak a little bit the protocol depending on the background of the pluripotent stem cells. But for actually creating genetic disease models, so we're going down that route now. Our initial push was to standardize the assay, which you would like to do with one cell line so it can be used as a standardized tool. We're now expanding out to recapitulate that with iPSC derived or patient cell types that have neurotube defects. We're also doing gene editing on our sort of workhorse cell line that we use for the assay in order to add some of the genetic mutations that correspond with neurotube defects as well. So we're going in those directions. We haven't done a ton of it yet.
Right. Have there been any findings that have surprised you in terms of screening for drugs? So, in other words, drugs that weren't suspected of having neurotoxicity in the embryo, but you screened, you found something, it was a revelation, maybe you even duplicated it in a rodent and found that, in fact, it really was a neurotoxic element. Any surprises there?
So we've had some surprises. We haven't gone as far as to do the rodent study yet, just because of the sort of immaturity of the technology. Basically, we just haven't had time to do all that. But it has become interesting when we talk to toxicologists. So when you find, and the EPA, I think, is now starting to publish some things with this as well. That these human novel alternative methods, right? That's sort of the classification that they're called. When they find hits that essentially did not show up in the animal model, which is what toxicologists have a ton of data on. How do you interpret that? And so there are some efforts to say, well, can you make your NAMs from the animal cells in order to prove that they do properly predict things, but it's really still an open question as to how that should be predicted, because of course there are significant differences in development between model organisms and humans, not just at the genomic level, but different cell types that arise, different structures that arise, different placement of structures anatomically. So it's still an open question. We've tried to find cases where there are, well-known epidemiological studies in humans, clinical studies that have shown that is a useful predictor in order to try to use that for our assay. You also run into sort of the pharmacokinetic aspect of it. So every compound is toxic at a different concentration, right? At a particular concentration. But is that going to be relevant for the exposure of, for example, a developing human in the womb? And so that's another area that needs more investigation to be able to better predict what are those concentrations that are relevant for looking for toxicity. So that's another reason probably why we see some toxicity in our assays that are never seen in vivo, because they're either metabolized or they never occur at that concentration in the actual developing embryo.
Great. Do you think your system could, in addition to looking for the neurotoxicity of drugs, do you think it's well positioned to perhaps look at various environmental perturbagens? So obviously the environmental perturbagens that are in our world, but maybe even some infectious diseases that are known to be neurotoxic, CMV, toxo, Zika.
Yeah, so I think Zika would definitely show up in our system. We haven't tried it, but anything that's going to affect, in our particular system, which is very early in neurodevelopment, so anything that's going to affect the formation of neural tubes, the neurotube structure in the early embryo, we think has a high likelihood of showing a hit in our system. And so that would include Zika, for example. It depends on where other viral pathogens are going to cause their effects. But if it affects the neuroepithelial cells, then we think it should show up in our system.
Yeah. And I guess the last question would be, is it your hope or expectation that this may become an FDA-approved assay?
Yeah. So that's the long-term goal. The road to take, right, is to try to make it something that's standardized. So that was our initial sort of effort, was can we say that this is not just a trick that we can do in our lab, but can we make this an off-the-shelf assay that we could ship to other people, have them do it themselves? And so that's what we first started to develop, precisely for going in that direction. Can this be a standardized assay where you can have the cells provided to you, you can have the engineered substrates, and you simply seed it into a well plate and run the assay? And we think that everything that we've done so far shows us that that is definitely possible. And so we'll continue to push in that route. And once we can, I guess, get the high throughput screening aspect of it done. So right now we're translating from a 12-well plate to a 9/6-well plate. Once we can get the 9/6-well plate done, and we think it would be very advantageous for presenting this to FDA, EPA, and saying, can we start going through the process of a standardized assay?
Great. One last question squeaked in under the wire, so I'll pitch it to you. Are you contemplating any epigenetic studies?
Yeah, so neurotube defects in particular have a very specific epigenetic etiology, and this is corresponding to the folic acid metabolism pathways specifically. So DNA methylation requires proper function of the folic acid pathway. And so we are thinking about looking at that as well when looking into the etiology of, when using the tool to screen for etiological factors of neurotube defects.
Well, this has been great, really fascinating, very promising, and an exciting new area for the regenerative medicine and stem cell field to explore, and reinforcing how important it is to marry multiple fields together, particularly engineering and regenerative medicine. So thanks a lot, Randolph, and congratulations on some great success, and we'll see you in June. Bye-bye.